Good morning, everyone. It's really a pleasure to be here today to tell you about the diagnostics-focused efforts that will be part of the NMIN. And we'll be switching gears here quite a bit just in terms of our focus and also the way that we think about bringing diagnostics forward that are really enabled by advances in nanotechnology. And I think when the proposal was originally submitted, the idea is that, therapeutics, we've heard about all the reasons why it's just critically important that we're there at the cutting edge developing new types of therapy, but the diagnostics have to come along, right, in order to really enable therapies to be used most effectively. So I'll tell you a little bit about the rationale behind this part of the NMIN and how we think about how nanotechnology can be leveraged to produce advances in diagnostics. We'll take you through what some of the challenges may be with this aspect of the network, and then we'll turn to telling you more about the projects that are going to be the initial ones that are funded. Here, we have a slightly different set of challenges than what you heard about with the gene therapy aim, in that the realm of applications for new diagnostics tools is very, very broad. And so we can think of many different types of disease and human disorders where patient care would really be improved if we had better diagnostic tools. I just list a few here, and you'll hear about the breadth of the projects that are going to be funded by NMIN. I think as the program progresses, we'll probably start to choose a focus and find the areas that we can really make the most impact, but we're starting out really leaving this wide open to really let the best science and the science that has the most promising commercial impact to kind of emerge and advance with the program. And so certainly new diagnostics for infectious disease is a very important area. We need to bring diagnostic testing out of centralized laboratories, enable it to be done right at the patient bedside or wherever patient care is being administered, so that we can limit the impact of infectious disease. Certainly in the diagnosis and management of cancer, right, we desperately need to diagnose cancer earlier when it's localized to eliminate metastatic disease. And then also, as we have more advanced therapeutics for cancer, we need much better diagnostic tools that allow them to be deployed in a way that they are effective. Many of the new therapies, as we heard in the last section, are incredibly expensive, and we have to target them at the right patient population. In the area of organ transplantation, this is another area where molecular level testing here could really improve our usage rates for donated organs, and so there's a lot of impact to be made there. And then also in rare diseases, some of the simpler rare diseases do have a genetic basis, and so sequencing can give you quite a bit of the story, but other rare diseases are much more of a kind of a phenotypic mystery. And so we need to come up with new diagnostic technologies that will allow us to get at the basis of rare diseases much faster so that they can be classified and eventually treated. And I could go on and on and on. Metabolic diseases, cardiovascular diseases, immune disorders. The spectrum here is very broad. I don't think there's any part of medicine that you can look at and say, oh, well, diagnostic challenges there have been solved, right? There's many, many unmet needs. And so I just want to say a few words about why this makes sense. Why is there a diagnostics theme within this nanomedicine NCE? For those of us that have worked on the diagnostic side, we've seen a huge acceleration in the types of technologies that are now being made available. And this has really come about because of the implementation of nanomaterials and different nanotechnology-enabled approaches. And really the way that the community has thought about this is just that we're pushing down in terms of fabrication capabilities into the nanoscale. We can basically make materials that have size features that are very similar to the biomolecules that we want to look at in clinical samples or maybe in vivo in order to diagnose. And being able to work in that part of the size scale has really opened up dramatic improvements in the sensitivity that can be achieved, specificities that can be achieved, and this is what diagnostic technology is all about. It's all about performance. And so I think that's how we think about this, that nanomedicine over on the diagnostics side is really nano-enabled analytical tools that allow us to reach some of these unmet needs that I described to you. And, you know, personally, just in terms of my own background here, I think this is a very powerful approach. I mean, there's this whole idea of shrinking down detectors and shrinking down materials so that they're at the biomolecular length scale. But what we found in my group is that this is actually a really critical thing to do to get good performance out of biomolecular detectors, because really interesting things happen at the nanoscale. And so for decades and decades, we were all working with these kind of flat bulk materials. And what you find is that if you go over to a type of architecture that has nanostructured features, that the biomolecular receptors that you usually use for detection and to do complexation at a surface, they actually function much, much better on nanostructured surfaces. They kind of have room to move. They're not constrained by a 2D surface. And so this is another just part of the rationale for really pushing the application of nanotechnology in this area. And so in theme 3, again, at the moment, this is a very broad effort. I think at the moment, our priority is on bringing in the best people working in this area in Canada to come in, make an impact within the network, help us really be able to realize some early successes. And then I think we'll, as I said, we'll keep focusing as we go. But I think some of the systems that you'll see are a focus and will be very powerful in the beginning are really seeking to deliver and be enablers of precision medicine, both in the context of in vivo diagnostics, but also in vitro diagnostics. And precision medicine here, I mean very broadly defined, just getting the right therapeutic to the right patient, being able to do very highly specific diagnosis of disease. There's also an emphasis on lab-on-a-chip systems as we miniaturize, right? That helps us get testing systems out of centralized labs and put them in doctors' offices or wherever they can be used closer to patients. You'll see that we are trying to develop an interface between the diagnostics theme and the other themes, and bringing in some of the lipid nanoparticle type systems into some of the diagnostic projects. You'll also see that there is an early focus on synthetic biology and really thinking about how we can use systems that allow you to produce biomolecular agents using synthetic biology really as a toolbox, and I think that's a very exciting direction for this effort. And so, you know, why is this possible? Why is this topical to do right now? Well, it's really the convergence of advances in nano fabrication and the ability to engineer nanomaterials, and our ability right now to continue discovering biomarkers and really match them very precisely onto specific disease states. And so I do think this is a timely effort, and we have the tools at our disposal to make a big impact here. And so many of the things that we'll do, again, this is a broad collection of things, but you'll see that there are technologies that are a part of this theme that are in vivo imaging technologies, and there's really a push to make these more rigorous and really have well-defined specificity, sensitivity, different types of performance benchmarks. Multiplexing is a big effort, to really do specific classification of disease. It's not usually a matter of looking at one biomarker, it's a matter of looking at panels of biomarkers, and so we need to leverage multiplexed methods. You'll also see some multimodal particles that help with that, also by allowing us to use different transduction types of techniques. And then certainly this point of care emphasis will come up again and again, and also keeping an eye on heterogeneity, which is really critical in most disease phenotypes, right? There's not just one thing going on, there's a multitude of things going on, and you need to be able to capture that in whatever biomolecular or cellular profiling is being done. And so some of our challenges, I mean, it kind of all sounds fine and good to miniaturize and bring diagnostic tools to the point of care. It's actually incredibly challenging for a variety of reasons. The regulatory aspects are challenging, the practical aspects of it are challenging just in terms of scaling up. And we know that the prices of many therapeutics are incredibly high, but the price point for many diagnostic tests is incredibly low. And so that presents a huge challenge just in terms of commercialization and also within the investment community and in terms of appetite for taking diagnostic tools to the clinic. It can be challenging to get people behind that. And so keeping an eye on practical types of miniaturization is really key. And a couple of these I've hit on: complex diseases, heterogeneity, thinking about in vivo versus in vitro and the advantages and disadvantages. Also, there's definitely a big push within the diagnostics community to get rid of things like the polymerase chain reaction, which is a way of amplifying whatever it is that you're looking for if it's a nucleic acid analyte. Certainly some of the advances in nanotechnology have allowed us to be sensitive enough that we don't need it anymore, which is good. PCR is almost too sensitive, and so if you're trying to run a PCR reaction outside of a really clean, sterile lab environment, you'll pick up contamination, and so it's a good thing to avoid. Many of the technologies that are part of Theme 3 are trying to get away from this. And so some of the advances that all of this is building on, some of them are really high-tech, some of them are more low-tech. And so we have increasingly sophisticated models that can also be used as diagnostics, things like organ-on-a-chip and organoids. And then we also have increasingly simple types of signal transduction and readout using things like smartphones. And I think it's a combination of those two types of approaches that will get us where we need to go in terms of making the right tools available. But I think in diagnostics, it's actually critical to always be as focused as possible on the simple things. What can be implemented simply, what can be manufactured simply, what's going to be cost effective. And so that's something that we need to keep an eye on as we want to support projects that really go to a commercial endpoint as quickly as possible. Another area, this is, as many of you know, an area where I work in liquid biopsy. This is it's a fairly new field. It's an emerging idea that perhaps we could get away from invasive tissue biopsy by looking at markers in the blood. And this is an idea that has come a long way. I mean, the term liquid biopsy is kind of a new term, but people have been looking at biomarkers in the blood for a long time. We do this using electrochemistry, which is a powerful detection approach for diagnostics. And so it's areas like this that we can continue to push and really think outside of the box in terms of how we can bring new capabilities to diagnostic medicine. This is another system on which we're kind of building on prior successes in the NMIN in the first set of projects. This is a little bit of a snippet from Keith Pardee's work, where he has engineered cell-free synthetic biology systems that allow you to create biomolecules kind of on demand using a cell-free type of system. And this is going to be deployed, I think, in a very powerful way in the first set of NMIN projects. And so then our 5-year deliverables, you saw some of these points earlier when Christian was presenting. And, we're really very focused on the outcomes here and what we're going to be able to say at the end of the day that we were able to accomplish with the level of funding that we access through this program. And certainly here it's all about commercialization. I mean, it is about excellent science, but that has to then be translated into something that has an impact commercially and has an impact on health. And so we are looking closely at partnerships between existing companies and our NMIN PIs. We also want to see new companies arising out of the science that gets done. And then certainly creating new jobs. I mean, there's a societal benefit here, but also certainly an economic benefit and the potential to be creating high-tech jobs through pushing this effort forward. So then, just a little bit about the strengths and weaknesses. I think, the great strength of the group that we have that's come together to work on diagnostic technologies as a part of NMIN I think the greatest strength is that we have a really tremendous community of scientists who are involved with this, and really all of the PIs that you'll see that are in that first cohort do have experience with commercializing new technologies, and so I think we've placed bets on a really good group of folks, and we do have that translational strength. And then the weaknesses, and I think things that we need to continue working on, we saw this during the application process, that there were some projects that didn't really think carefully enough about the complexities of looking at clinical samples and dealing with things like biofouling. But I think maybe even more of something that we need to focus on is just a lack of coherence. So you'll see that we've funded a set of, I think, really excellent projects, but it's hard to say that they're all really kind of focused in one area or doing something where there's synergy between the projects. And so we have to continue to work on that, and certainly we would welcome your feedback. So with that, to get into the specifics, I'm going to turn things over to Gilbert, who can walk you through the round one projects.
So I want to add one point, which is the weakness, which is this biofouling problem or nonspecific binding and so forth. I want to emphasize that one of the examples that came up in the seminar yesterday was the use of the exchange of a ligand for a nanoparticle with a protein that actually helped this thing to get into hepatocytes, right? So it was the transition of the material that enabled the function. And one thing that's really exciting about what's going on in this network in this particular theme is an effort to learn more about what goes on to particles in order to exploit that kind of a phenomenon. So these are the things that were funded in this, or are deemed eligible for funding. I guess they're all sort of in a liminal state, to use a good anthropologist word. And I'm going to tell you a little bit about each of them. They are in sort of distinct areas, as you'll see. Most of them, in fact, somehow or another integrate liposomes and have applications of the liposomes. That's definitely something that's viewed as a rich and robust technology by the network, and there's a sense that those things should be adopted, and also there's a sense that down the road, one of the ways that the different themes can integrate, for example, the way to have better integration among the theme 3 and the other ones, is to provide characterization of those materials in a way, and a characterization of them as they go through the body. That's really what, it turns out that the majority of the people in the theme 3 are somehow analytical chemists, actually. And that's the kind of skill set that they bring to the table that I think will be valuable. Of course, everything is nanostructured. And since they're all chemists, actually there's quite a strong ability from this subgroup, to also help other people in the network with that stuff. There are a few technologies that stick out as being used heavily. Mass spectrometry just happens to be one of them. The connections are, as I said, here through this, the liposomal stuff. But I also think that the strong kind of connection that's really not made as well as it could be made is to have diseases that are well represented or carried in effort between the different themes. At the moment, that's not really done very much, right? There are examples of specific diseases that you'll find in each of the different themes, but there's that are the same in each of the 3 themes, but that there's not as much cooperation or strengthening as there might be. So that's, I think, a place where this could really be improved. Whoops. The cores are very essential. The cores you heard a lot about in the last, not Shayna's presentation, but Peter's before that, and also Christian's. This theme uses basically mostly the nanocore, but also uses the pharmacore. And I'm going to tell you a little bit about how that works. There are people who are both kind of users and really experts at this. So Warren Chan, whose project I'll tell you about, expects to use the PharmaCore, of course, but he is a deep expert in this area, and I hope will actually bridge some of the connections between the NanoCore and the PharmaCore that will enable some new technologies. And I'll tell you about these other projects sort of one by one as we go on. There is no core in the same way for theme 3 as there are for, in some sense, for themes 1 and 2. Actually, the core concept came up a little bit late in the game as we developed the proposal, as we thought about things that were legacy opportunities. And I think that's actually something that, as a group, we'll probably come up with our other ideas as a function of time about things that we want to really build on that will become standards for certainly for Canada, but maybe more broadly for the world. So in the biodiagnostics, there's a theranostics effort as well. So making particles that are both drug delivering and for example, labels for detecting the performance of detecting the location of the drug and ultimately biodistributions. So this is an example of a project from Gang Zhang. This is a project. He has a lot of expertise in integrating liposomal materials, for example, porphosomal materials, and also radiolabeling, which has great diagnostic advantages over and analytical advantages due to quantitation over other techniques. And he's integrating this with a nanotexiferin system as one example that he brought up. The stuff that has been going on in Shayna's group, she mentioned briefly, has to do with exosomal analysis for cancer detection. There are a variety of different ways that exosomes contain information that can be useful diagnostically. I mean, of course, people analyze proteins, microRNA, messenger RNA, and things that are like that inside. Very often, there aren't the correlation of the species that are present, mRNAs versus proteins, in exosomes is not uniform. And I think there's some really interesting opportunities as we begin to develop data on the cross correlations that give us, identify for us tracking mechanisms for the performance of the materials. Because you talked about this at length yesterday, I'm not going to, or parts of this yesterday, I won't talk about this much more. I'm really, really excited about stuff that's going on in Keith's group. The whole concept of precision medicine requires that we be able to develop, and personalized medicine requires that we be able to develop delivery systems, you know, on the site, not send it off to some company to be developed and then come back, you know, a month later or something like that. So the ability to have a toolkit to create functionalized materials for targeted delivery or for appropriate packaging of the carrier and preparation of the carrier and packing of the drug is really, really, really powerful. And I'm really excited that Keith has been drawn into this. He's tremendously busy, but this is actually turning out, I think this has one of the deepest promises among the things we have. I can think of any technology that's going on anywhere in this net, or many technologies within this network that could benefit from this particular thing, and so we want to support that definitely. So there is work done by Warren Chan, which used to be called opsonification, I guess is what it was called, or the building up of protein coronas around nanoparticles or other kinds of macromolecules, which I was trained because I came from a sort of an engineering committee in the back of this is kind of biofouling. But really, the kinds of things that he's trying to learn is what proteins build up on particles. He has early examples, really excellent examples for the buildup of proteins from serum onto nanoparticles, and that's been really focused on metal core particles, and then trying to figure out how this affects the biodistribution and cellular uptake, and then to try to develop predictive models for how different surfaces absorb different proteins and thereby guide where those nanoparticles go. This is the focus in his lab in the recent past has not been so much on liposomes, but it will be significantly on that in this network effort, which I think is really great. I think it gives us a wonderful way to integrate new information into for example, NanoCore, a set of predictive aspects or predictive models for biodistribution, having analyzed the proteins that build up on different nanoparticles under different contexts. He's developed pretty complicated and sophisticated tools to do that. Accumulation, half-lives, things like that. Okay. In my own lab, we're developing a device that will allow us to look in a single cell level of the interactions between pairs of cells, phagocytes and target cells, which is a microfluidic type device, which integrates chemicals designed to alter the interactions between those two cell types. And so this is an example of something that may find a way to integrate among themes. Our goal is, of course, with such a platform, to be able to use it as almost a drug discovery platform, as a way to find to evaluate interactions among different drugs and different adjuvants, to try to see if there's ways that we can optimize or find screening done more rapidly. So when I was asked to talk about NanoCore, of course, since I don't work in NanoCore, I had a different perspective. My perspective was I want to use NanoCore, not I want to be a NanoCore provider. If I, as a user, there's some things that I would like to know when I access this, and that's kind of how I looked at this request from Diana. Why do certain compositions work into certain contexts? Why did the in vitro work not translate to in vivo work? And what kind of algorithms were used to generate these libraries of nanoparticles? Was it really one by one? How was it a series of physical properties and one scanned a particular composition vector and then the other one? How were these things generated? That will help us to understand what's maybe not in the library. And other technical things that are more selfishly inclined, like different kinds of drug loading versus compositions of lipids and cholesterols. So finally, tutorials on lipid, the toxicity of lipid nanoparticle or liposomal nanoparticle compositions. So these are what the naive person asks for. And I think I'm hoping that these will be the kinds of things that we might be able to get access to. This might be a tutorial opportunity. This is the kind of thing that the network is supposed to offer. And that may be a more rapid strategy than doing one-off with each person and teaching each person what they need to know. You could also guide us individually to a place where all that information where a lot of that information is present and get us started. Because we hope to have an efficient process here, because we only have a pretty short period of time. These projects that are slated to be funded are 2-year projects, and it's expected that they will lead to after a year, they'll have kind of go, no-go points. That means that if you haven't met the metric that you stated at the outset that was accepted by the review committee, then the money is stopped. And why would we do this stopping? Why would the network stop the money like this? Well, because we're supposed to be producing things that can be taken up by industry. And the pathway to clinical development is different in biodiagnostics than it is for the drug delivery or the gene therapy. Our goal is not so much to get things into animal models at the beginning, which is one of the key goals for themes 1 and themes 2. Our goal is more that we should be using our diagnostic tools to test and to analyze human samples. as opposed to and samples that are clinical samples in that sense. And then we have other metrics which are of course related to efficacy and so forth. And so one of the things that we try to emphasize is that people should develop a pathway to their product in the diagnostics context. This is an example of how it would flow. Again, to generate a technology dossier that will be attractive to a customer. The customer would be a company or maybe a spin-off company that you would have yourself, the investors for those folks. So the pathway is different, but it still involves getting deeper connection to the human samples that are critical in the clinic. So now I'm going to open it up for questions and discussions and ask Shayna to join me at the famous station here. And be very glad to hear your comments and questions. So thanks very much.
One of the things that is for me as an outsider in the area of diagnostics is there seems to be so damn many of them, and they're all over the place. And how is it that we could maybe within the nanomedicines network get them so that they really do make it all the way through and really enhance that probability that they're going to be successful products?
Okay. I was hoping that was a question for Gilbert. It is a good question. I mean, talking about thousands and thousands of papers, right? I mean, in the analytical chemistry community, we have hundreds of thousands of papers on new diagnostic technologies, but not much has changed. And I think it really is about coming up with systems, I said this, that are as simple as possible, scalable as possible, and where you choose the right application to commercialize with. And trying to go out with a new technology where the test that you're trying to offer is something that the healthcare system will only pay $25 for is not a good idea, right? You'll just never get there. It sounds good, right? It sounds like a good idea, but you just can't get there. So I think being very strategic about not just what are the bells and whistles that a technology has, but what is it being applied to, and how are you going to get there? I think that whole process has to be looked at. I mean, we've funded a small set of first things that were kind of in the original proposal, but I think as things mature, we need to look much more at the practical aspects of this, because the bottom line is that you can have something that works amazingly well, but if the other elements aren't there, you're never going to get an investor behind it. You're never going to get a strategic partner that's going to agree to take it forward. It really, it has to hit all of those practicality benchmarks, because, again, it all comes down to money. I mean, if you can charge $1 million a dose for a drug, you're going to get away with a lot of things that are really difficult to implement, for better or worse. With a diagnostic test where maybe it's going to be $25, $50. If it's a really high-end thing, it's going to be $1,000. But still, the economics are out of whack. So I think we have to keep an eye on that.
Thank you. We have a question over here. Please introduce yourself and your organization.
Sure. So John Walmsley, I'm with Starfish Medical. Those of you who know us would know us as medical device technology and product development consultancy. And where I think I'm interested in a continuing conversation in this specific area. What we like to do is take people's great ideas and commitment to taking a product to market and provide the technical and engineering support that gets them there. And I don't know if you know, we're headquartered in Victoria. We have an office in Toronto as well. And we're just in the process of instigating microfluidics, scale-up manufacturing, design, development, and manufacturing capability, and really interested in helping this group work through the challenges you were just discussing. So we have something that works in the lab, and how do we make this work? You go through the process of is it even possible, and then is it likely, and then is it certain? And we're really in the moving it from is it likely to it's certain business. So I think I'm just making a statement that we're interested in that conversation. I'd be interested in any feedback, but thanks.
Marcel has a follow-on question. Thank you, John.
Well, it's going to be a little bit different, I think, and you'll hear a cautionary tale when I talk about theme 1. And I guess it's related to, on a positive note, that you're mentioning the KISS principle, keep it super simple. That's the goal to get something into the clinic, so you have to have that every time. And I have been involved with a number of groups that have done diagnostic assays more simple than what you're talking about. And they fail often because they lack specificity and what's the other one? Sensitivity. And they don't meet the criteria that they must meet. And now that doesn't necessarily stop a paper from being published. You can still publish it. So I guess the question is, how, as a group, are you going to address that particular issue? Because it seems like that's the issue that needs to be overcome to be able to be commercialized.
Yeah, I think
So there are pretty recognized standards to sensitivity and specificity that are in the business, and I think both Shane and I can see those at the time. What I might suggest is that an interesting way to address these is to think about diagnostics or tools that can be used specifically with the kinds of diseases that we're trying to target and the technologies that we're using to evaluate the performance of those therapeutics and so forth. That might actually be a way to also develop. In many cases, that's not necessary because it's quite obvious what the outcome is, and you don't actually need such a follow-up diagnostic or evaluation. But I think there might be useful feedback on the simplification demands by interacting with folks who are carrying these things further down the road. I think there'll be a force, right? It's like whenever I talk to the clinician I work to, he always tells me to simplify, right? And so I think that would come if we partner some of these technologies across themes.
I think also there has to be an absolute requirement for working in complex samples. And I mean, all the analytical journals are going this direction now. I mean, you can't publish a paper on a new technology unless you have gone out and found some serum and tested for your biomarker and not diluted it 100 times. You know, you really have to show that the performance is there. So I think programmatically we can also insist on that as being the go, no go. You know, does this work? Because it is important to fail fast in diagnostics, and if you have something that is just too prone to fouling, just fundamentally the sensitivity isn't there, I mean, you can go back to the drawing board and maybe try to solve the problem, or maybe you should just forget it and go try something else. And so I think we need to have that fail fast principle embedded. And I also wanted to touch on the comment from John at Starfish. I think in terms of really assessing practicality of the things that we're doing in the diagnostics teams, I think you guys could, I mean, working with you would be tremendous in that respect. A lot of what happens, I mean, maybe once you do get through that gate where you've shown that you do have sensitivity and specificity and you can do what you need to do in patient samples or even in an animal model, then typically what happens is there are cycles of design, and you kind of go through the design process over and over and over again to get to that clinical grade prototype. Doing that once instead of 3 times by working with a Starfish or an outfit that really understands all of the quality regulations and all of the things that need to happen in order to get something into the clinic, I think that could save us, right, from just getting stuff. You know, there's a certain kind of valley of death in diagnostics, and a lot of the design cycles and that kind of stuff is part of that valley of death, and that will definitely help us to partner up.
Great, thank you, Shana. Other questions or comments? So, John?
I could just shade a little bit on the sensitivity and specificity. So there's a very high bar if you're going to make a diagnosis from your data, but there's a range of bars on that if you're providing extra information to someone else who's going to make the diagnosis. So it does need to work, but you can get better over time by modulating the claims that you're making of your device, of your system.
Yeah, I just want to put Mike Cochtery on the spot. From a pharmaceutical science or faculty pharmaceutical science point of view, obviously there's an increasing emphasis with things like companion diagnostics and so on. Whereas most of the effort, of course, is in making new drugs or using already approved drugs more appropriately. Where do you see the trend lines going on the diagnostic side?
Well, I mean, I think I'm not really the expert in that personally, but I mean, I think it's clear that identifying the really top reliable biomarkers to turn into diagnostics still remains the gold challenge. I think that everybody faces. I mean, turning something that, I mean, look at PSA, for example. I mean, turning up something that's much more effective than that as a prognostic or diagnostic marker. Those are the challenges, and there's a lot of work going on. Obviously, the technology has advanced to the point where it's considerably easier to look for biomarkers in biological materials, but the challenge still is translating that into something that can be used effectively, readily in the clinical setting. And the big challenges are probably in developing. How do you translate something like that into a developing country where there might be a really widespread need for it, but not necessarily the technological or infrastructure to set these things up. So I still see the translating that into a reliable diagnostic test or whatever it is that can be used widely without necessarily access to high technology infrastructure. Those are the things that I think that are probably going to have the biggest impact worldwide.
Okay, thank you. Any other comments or questions? I know, Gilbert, in your presentation, you talked about Gang Zhang's project, and I actually think we have 3 trainees here, the lab manager, Lily, and Wes and Alex. You're from Gang's lab? Right. So do any one of the 3 of you have any comments about that project in relation to Gilbert's comments in terms of presentation? We want student participation too.
Sorry, which comment in particular?
I'm not actually exactly sure, but I think what she was saying was, do you want to say what he's doing? And maybe the answer to that is no, which is a good reason to say, what's the question again? So let me turn the question around and ask, so I don't know what stage of education you are in these, but what kinds of things do you want to get out of this network that you think we ought to provide? Let me change Diana's questions a little bit.
I guess like the industry aspect of this is really interesting, especially with the Metallo Nanotexafrin project, because it's a platform. The nice thing about Texafrin is it chelates a ton of metals really efficiently. So it's important to kind of narrow it down to which metals or which radioisotopes have the greatest impact and are more likely to be translated. For example, we could chelate it, like currently in the application we submitted, it was lutetium-177, because it's got a lot of applications in SPECT imaging as well as radiotherapy. But you could just as easily chelate it with indium-111 for higher SPECT sensitivity and Auger electrons, or you could use manganese and gadolinium for MRI contrast. So I guess just in general, kind of understanding what the focus is and where the clinical need is and how we should be aiming the project going forward is important.
Yeah, okay. So one thing I've noticed a shortage of in this room is clinicians. Maybe we have one clinician, I'm not certain. Maybe I don't know everybody.
How many clinicians do we have in the room? Just raise your hands. Hands or hand?
One.
Yeah.
Sort of.
Yeah.
Well, Alec is a medical student. He's doing medical. Sorry, say that again with a microphone so we can hear.
Oh, I'm in the Department of Medical Biophysics at U of T.
Right.
Right.
So thank you. And we need more like you, I think.
Over here.
Great.
Blair.
Blair Levitt, yes, Blair Levitt as well. Sorry, you're behind somebody, so I was.
Yeah, Blair.
Okay,
Is there
great.
anyone else who's the clinical background? Okay, so we've got 2.
I think that will add a great element to the conversation. So, but we'll see how that goes.
So that's a shout out for everyone to try to find a clinical collaborator on your project.
So I think
your
actually
homework.
many of us have clinical collaborators. I certainly do. They are tremendously busy, and to ask a clinical collaborator to come from a significant part of a day is a gigantic demand, especially because he actually is constantly trying to solve problems to keep people healthy, and that's his highest priority. So, but to the extent that we can encourage people to come, that would be really great, and I'd like to figure out how to do that better. Maybe if we can encourage people to think about that a little bit more.
Right. Alex, did you want to speak? Okay, we've got someone up here. Okay, Michael, and then we'll go back to Alec after Alex after that.
It's just actually a follow-on. It might be worthwhile presenting or getting a booth at something like the Society for Nuclear Medicine, because I think, PET and SPECT
Right.
are clearly technologies where I think nanotechnology could have a tremendous influence. So I know they have annual meetings, and might be worth
Right.
getting one of you guys to be
Yeah.
invited or to have a booth at
Yeah.
one of those
Yeah.
society meetings.
Right. And yeah, thank you.
We
Good
have
suggestion.
we have a bit of a speaking list here. Alex, and then you're next. Alex?
Thank you, Diana. Just to build on what you were talking about earlier, Gilbert, with regards to building a reference system or a base of tutorials for people within here, if as PIs you think it's a little intimidating to become completely familiar with the vast body of work here, speaking both as a medical student and a graduate student, I can reiterate that fact that trying to condense and distill the most important aspects behind the vast amount of research in nanomedicine, I think is very important, and I think that NMIN can really provide an instrumental role with regards to that. With concerns to my clinical training, there's definitely a lot of excitement about what nanomedicine may be able to do, but the exact details on how it can provide advantages are still a little fuzzy, and there's a lot of hearsay, at least at the early stages of clinical training. So ensuring that there are readily available and accessible resources would, I definitely think, help with bolstering those basic science to clinical relationships and understanding that if they are to be building those collaborations, how can they make sure that everyone is on the same page and that everyone is providing equally to enabling that translation?
Yeah, being able to explain it in a brief elevator pitch is a great skill.
Alex, thank you for raising that, because it really does loop back to what Peter Cullis was saying to us this morning, that communications is an extremely important, I think, responsibility and role that all of us in the network can play. And perhaps what we can do at the Administrative Center with some help from the experts is actually develop sort of one-page case studies of things that worked. Peter talked this morning about things that work remarkably well and were approved by the FDA. And I think we all need to be ambassadors for the things that work and be able to talk to people about how this works well and why this is the next big thing in health, in healthcare and health diagnosis and health treatment. So, illness treatment. So I think that comment's extremely helpful, and we'll feed that back into our communication strategy within the network over the next 6 months. We had another comment here. Yeah, and introduce yourself to everyone. And
So I'm Naomi Metzer. I'm from the University of Toronto. I just wanted to alert this community to the fact that there is other pan-Canadian networks, for example, in molecular imaging. There's, for example, the World Molecular Imaging Society just created a Canadian chapter this year. So it's there's people from UBC, people from Montreal, Toronto, London, whose expertise is on preclinical and clinical imaging, so on the research end. So that might be a network where if you need some imaging expertise or you want to do some particular kinds of imaging of your nanostructures, that group is certainly a very well-connected group, both clinically and preclinically, if you'd like to try something out. So I could connect you with that group if that's of interest.
Absolutely. So send us those links.
Okay.
And this is now a shout out to Marshall, our webmaster. And we will make a place on our website to put those links there for easy access for everyone, should you be looking for collaborations, partnership, or networking with other networks. So that's a great suggestion. Any other comments? Yep, at the back here.
Hi, I'm Wilson from University of Toronto, Chan Lab. I had a comment from just hearing what Alex was saying here. So, is it true that sort of like for medical students, there's not sort of any course or introduction or formal introduction to nanomedicine or next generation medicines in general?
There is one taught as a team taught course at U of T, organized by Gang Zhang, and there are about 5 of us who co-teach it. But it's one course. I don't think it provides as much information as we might like it to.
So that's only for medical students. I'm just wondering. No.
Oh no, that's not. No.
Okay, because I'm in the Department of Biomedical Engineering, and I don't think there is a course that we have currently, at least. And I also was previously at McGill University, and there was a course on sort of micro nanotechnology, but the focus was mostly on microfluidics and not so much on nanomedicine. So I'm just curious, since we have so many faculty members here from across the country, are there any sort of formal nanomedicine-based courses that are available to students? And maybe should the NMIN sort of, I wouldn't say standardize it, but at least provide some sort of resources so that we could actually train students and medical students in it more in a more organized manner? I was just wondering, what are your thoughts on that?
All right, so
Star has some comment, I think. No?
Yeah, Star and then Marcel, but just to.
I was going to redirect to Star.
Okay, you're redirecting.
Star Lee from UBC.
Good SWIP.
Faculty of Pharmaceutical Sciences. We do have a nanomedicine course offered at our faculty. It's called FAR 515. It's co-taught by many of us sitting in this room. So basically covering all ranges of nanomedicine, nanotechnologies happening on the campus mainly, but also being developed around the world. So very cutting edge research topics.
So Star, is that something, Wilson, you're at U of T, right? Is that something that we could leverage within the network and perhaps make it into a modularized series of webinars for everyone in the country?
So I think we definitely have the capacity to do that with our facility at our faculty.
Right.
Yeah, but we can definitely look into this opportunity.
Okay, terrific. Thank you. There was a hand up. We've got Urs, and then we've got, I think, Naomi, and then Michelle.
Okay, it's Urs here from UBC. So I also teach another course in our pharmaceutical sciences program, and it's called Diagnostic Imaging and Radiopharmaceuticals. And the aim is actually to find out, to compare all the different diagnostic possibilities for nanomedicines and then choose the right one, And so we do that every year. It's a 2-week block course. And I started that because I'm in Copenhagen a lot. And I have lots of people actually from Copenhagen that come and take this course, So that would be open to everybody here if you want to do that. And I have the program ready, for next time. So if you are interested, I can send you that.
Okay, send that over to us, Urs, because that sounds very interesting.
So it's a graduate course.
Right. Okay, and then we have, next is Naomi and then Michelle.
Okay, I just wanted to alert Wilson, you to the fact that Christine Allen, through this network, has delivered a nanomedicine course 2 years ago through the pharmaceutical science department. So that has been going on. I think some of my students took it; they really enjoyed it. So there is that going on, and perhaps we're not marketing it well enough. People within the network don't even know that these courses are going on through NIMN at the University of Toronto. So.
Okay, great. We'll circle back with her. Michelle?
So we have a course at Polytechnique on biomedical nanotechnology, more on the engineering side and physics. And This is taught in French now, but it will be in English next year,
Okay, great.
with
And-
labs and so on.
What's French for nano?
What?
Sorry.
All right, so the speaker order is a little bit confused. We have, who's got the microphone? First
okay,
here,
yeah.
and then we'll go to the back. Yeah, Rob, you're next after this speaker.
Yeah, I'm Azita from University of Saskatchewan. We also do have a nanomedicine course. I teach I have one course for graduate students. And then in that course, I talk about different types of nanoparticles, and then it's more kind of related to the students that are taking the course. So what their project is. So it could be more into the preparation, different methods, how to make it, or more into the medicine type of it, targeted delivery. And so it's kind of, I always offer new topics, but the course is nanomedicine. And also I have vaccine delivery, which is more into nanoparticle, vaccines. And then we also we have a course in undergrad level for the PharmD and also bachelor students. We do have kind of advanced drug delivery systems incorporated in our curriculum, and we teach for different oral delivery systems and also IV. So we do have that.
Okay, that's great. We got a wealth of opportunities here. I'm going to go to the back now with Rob Sutton, and then I need my team to sort out the speaking order because I've lost track. So, Rob.
Well, it's Bob with IDT again. We, of course, don't offer any faculty courses, but we have a wide range of educational tools on our website for training in cargo and nucleic acids and what you need to put in there, how they work, how to design them. The design tools are free. All those things are there. Through the years, we've done a lot of modules in fourth year graduate level courses for a specific day or two on nucleic acids, how to use our tools. We also do a lot of symposia collaborative work, so I would offer those sorts of collaborations to any of you who are doing classes. If you want specifically targeted lectures on cargo, siRNA, CRISPR, any of those sorts of things, and the functionality, we're happy to participate and join in. And the same I would offer to any of the graduate students out there. We do a lot of local symposia and support those, and I think that's one thing we can do with the network is help to bring a lot of that together on a very regional level. So I would also offer our collaboration to any graduate students who want to do half-day symposia, seminars on cargo, on nucleic acids. We're here to help, and we can really provide a lot of support there.
That's great. So do you want to give a shout out again to the web link that people go to for the information you just described?
Certainly, yeah, I can find out links for that.
Yeah, but it's IDT is the company.
Yes,
IDT DNA. Okay, that's terrific. So there we go. We've got an HQP partner out of this conversation. Thank you, Rob. Okay, team, who's next? Alex.
Hi, just to iterate on Wilson's question about on the pre-clerkship medical side of things with regards to nanomedicine teaching. Out of 2 years of pre-clerkship, there's probably less than 2 minutes total discussion time devoted to the idea of nanomedicines, and it's almost completely just about liposomal formulations and doxil. And so really distilling down what might be the most pertinent factors that you would want to pass on to a early stage clinical trainee audience would be very important. Because there is both a balance between the most pertinent details that you want to pass on, but also the level of comfort with the clinicians which are teaching those courses with regards to how they may be able to pass on that knowledge. If the clinician isn't particularly comfortable with describing nanoformulations, then in all likelihood they'll choose not to focus on it. And so with just based upon that perspective, it's great to hear about all of the resources that are available for graduate students, but in terms of enabling clinical relationships, making sure that you're bridging that gap is very important.
Great. So if we're really going to make a difference in the clinical setting, we need a two-track strategy to educate the clinicians and the clinicians that are training the clinicians. the trainees and the clinicians themselves. Okay, fantastic. Great advice. Any other suggestions, ideas, gaps, silences? Is there anything you've been thinking of? We've got 4 minutes left that you want to give voice to here. Somebody's got the microphone.
Okay,
Go ahead.
I grabbed the microphone before that, so it's not necessarily answering your questions, but
That's okay.
I'm Mike. I'm a grad student in Christine Allen's lab, and I was just going to build a little bit on what Naomi said earlier. And Christine did teach her nanomedicines and oncology course. I think it wasn't offered through NMIN. It was the NSERC Create program, POND. I think we saw someone yesterday from Matt Moffat's group talk about that a little bit. It's like a polymer drug delivery network. I know that she found it very difficult to sign up people from it was offered to people at other universities throughout Canada. And I know that U of T made it rather difficult, I think, for them to register in the course. So it's something to think about if you're planning on actually registering and have people take the course for credit. And that was at the graduate level, I think. But she did teach it to the undergraduate pharmacists and pharmaceutical chemistry students as well.
So it's it's a course you can get a U of T credit from.
Yes.
Okay. Not necessarily recognized by your university issuing your degree if you're not a U of T student? Is that what you're saying?
No, I mean, the students from University of Victoria and University of Montreal, I think, did get credit for it ultimately, but their institution seemed more open to that system of taking courses remotely than U of T was. But.
Right. Okay, great. Thanks for that, Mike. Anything else? Yes.
So I just want to, I guess, bring a conclusion to this discussion topic. But one thing I just want to add is I don't know if all the students in here have the same opinion as me, but I found that when we did take some of these sort of cutting edge, for example, nanomedicine lectures that were co-taught by a lot of different profs, it just turned mostly into a buffet of what the individual PI was interested in doing. It just basically showcasing what their lab was most interested in. And while that is very interesting and very insightful to what is actually happening at the moment, I think a more useful approach would be to have all the fundamentals and all of the core techniques and analytical methods and experimental methods taught first, and then maybe move into sort of the cutting edge after. So that's just sort of maybe my opinion on this.
Right. So set the foundation and then build on that. Thank you. Well, we've got
Okay. First, I just wanted to say that it was a great suggestion, Wilson. I think that's really good. I really like your ideas on that. But I wanted to change the topic. Maybe something Gilbert a little bit alluded on is just another gap that I feel is here. I don't know, maybe you have strategies to address that, is to find synergy between different teams, in terms of the research. I see that there are lots of potential for collaboration between different teams, and then if you put 2 and 2 together, maybe we get 6 or 7 rather than 4. And so I wanted to know, are there any plans or strategies to make that happen, this synergy happen between different teams? Like, for example, diagnostic can help promotion of some of the, for example, targeted drug delivery or the gene delivery teams.
Maybe I'll take that question just in general. In general, because job one is to get the funds that are in our account here at UBC out to you folks to get doing your work. And just to remind you, the effective date for your use of those funds is September 3rd. You may not get those funds until October 3rd, hopefully before that, but you can start to work on your projects as long as you can move some things around and then reimburse yourself once the funds arrive. But as a general rule, once we give you an opportunity to do your work, we're also going to be planning to have research workshops, theme-based, opportunities to talk to each other and work out any kinks in your projects with the team, but also then engage in facilitate and enable cross-theme dialogue and discussion. Because from the government's point of view, the NC is one big project. Now, right now, we have 20 funded individual projects and 3 themes. So we've got some, what I call knitting the network together to do, and that takes time, and people have to talk, and people have to work on their projects. But, taking into account everything that we've discussed over the 2 days we're together here, I think people will approach their projects with those considerations in mind about, linking with other things and how do we align things to have a greater impact working together than just working as individuals. And so we'll have those events over time. There also will be an opportunity for theme-based teleconferences, but that's something that we will be doing, and there's mechanisms to do that, but it requires us to, have time to do your own research, but then come together and have those conversations about, where are we and where are we going? So it's definitely in the game plan, and thank you for raising that. So I'm going to turn it back to Shana and Gilbert for final comments before we wrap up before lunch.
Just to reply to the question that was just made, actually in the proposals that we have, there are many that exhibit people who I will call, whose main footprint is in a different theme than the leader of a given proposal, and that is a way that I think integration is going to naturally become enhanced already. So I think that will work. I'm just really glad, especially to hear from trainees. That's, I think, what I want to say here, because that's at least half of why I'm here. And so I'm really, 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 really thrilled that we've had some input. So thank you.
And just to add to that, I think as the network evolves and matures, there will be, I think, project partnerships that come about where we are trying to take 2 and 2 and make 6 or 7 or whatever. And I think it's great for all of the PIs to be in one room to start thinking about how we can make those linkages. But to piggyback on Gilbert's point, if you're a trainee and you see another project that you think there is a link with what you're doing, then try to make those linkages trainee to trainee, and then let the science kind of percolate up or to the sides or whatever. And we definitely encourage, the ideas that get pursued here to not all be kind of, PI-led types of things. So I think it's important that we're all in one room to let those synergies kind of arise, but they should come from really any level where there's inspiration.
And so just to follow on to Shana's comment, and I don't know if Leah Greystone, our HQP coordinator, is in the room with us at the moment. There she is, Leah. We're just, I think one of the options we haven't framed that HQP capacity building program yet, but the networks that we've worked with previously always had trainee lab exchange funding to enable trainees from one lab to go and do a visit to another lab and vice versa. So there's these reciprocal training exchange opportunities between and among labs in the network, within themes and across themes. So, and even international opportunities to travel to another lab if it's justifiable to bring back some knowledge, skills, or insights that are relevant to the NMIN project. So, not making any promises, but I think that's definitely a viable option to build on the observation that Shana just made. So please join me in thanking Gilbert and Shana for a tremendous presentation this morning.